That
There we
is
go.
very far away from us.
Hello there. This is a recording for the Body Mind Centering Study Group follow-up. This is today's date is April 4th, 2024, and we're reviewing the brain development. So wonderful to have you guys here. And I picked up this wonderful video that I thought would be helpful to look at so that we understand more clearly some of the concepts that Bonnie has been presenting to us. I'm going to turn off my headset because the sound will play better from the video. So let me put it on share so you can see what I'm seeing. And this is development of the nervous system, embryology with Dr. Mike. So we'll start here. And put your thumbs up if you can hear him talking. No. Okay, so here we go like this. So now I'm turning it off and we'll start again.
This particular lecture, and the first being the formation of the brain vesicles and the folding of the brain itself. Number two is how the neural tube.
Okay, so we didn't get the very first
short lecture on the embryology of the nervous system. So in this particular lecture, we will look at the general development of the central and briefly the peripheral nervous system. There are kind of three main learning outcomes for this particular lecture, and the first being the formation of the brain vesicles and the folding of the brain itself. Number two is how the neural tube forms and causes the creation of all the cells in particularly the central nervous system. So that's almost more like histology of the neural tube. And then finally, we'll finish in number three, being the positional change of the spinal cord itself. So let's go across to this image. Now this.
Okay, does that sound okay for you guys?
Yes.
Yeah. Okay.
These two images we used in the introduction of embryology or the embryogenesis. So if we look firstly here, now this is about 3 weeks. We've got the top blue, which is the ectoderm, the underneath black, which is the endoderm, and then we've got the mesoderm now filling up in the middle, which you can see here as well. So it's coming in from the epiblast in. Essentially, the epiblast will make all layers, but in this case, we've got the 3 layers in place now. So the mesoderm is now filled up the middle, so you've got your 3 germal layers intact. Now, this is cutting through in a cross section, and if you were then to fold it back and look down, that's the view we've got here. So we've got the endoderm underneath, endoderma here, going down to the yolk sac, going down to the yolk sac, and then the ectoderm reflecting up like so into the amniotic cavity. So towards the end of the 3rd week, we've created the mesoderm, but what happens down in the midline, right down the middle of the embryo?
Okay, just want to check in with you. Any questions at this point? Are you guys? Okay.
Yes, I have a question. In terms of his drawing, on the left bottom.
Yeah, back
I
to,
lost it.
yeah, coming back. Let's see. Yeah, I would go ahead and say that and I'll find this. Or so you need to have the, why did I put it like that? Go share portion of the screen. It's there. Let's go back to.
Yeah. So the bottom left, he has.
This one?
Yeah, so caudal end is on the right.
Yes.
And the cranial is on the other side, but
Yeah.
there is on the blue, is that the blue is a neural tube?
The blue.
Yeah.
Let's just hear him speak about it again. I don't want to
Because there is a cut there, and I am wondering why is the cut there between
Well, he was
these.
talking about the epiblast.
Okay.
Epiblast producing these layers. And I thought they was meant here that the epiblast was shooting the layers in from this opening.
Okay, and what is the epiblast? Sorry, it's a,
That's the source of the new cells that produces the 3 layers, if I remember correctly.
okay.
So we'll go back and just look at that for a second.
3 germal layers intact. Now, these 2 images we used in the introduction of embryology or the embryogenesis. So if we look firstly here, now this is about 3 weeks. We've got the top blue, which is the ectoderm,
Okay.
the underneath
Ectoderm.
black, which is the endoderm, and then we've got the mesoderm now filling up in the middle, which you can see here as well.
Okay, so the blue is the ectoderm.
Okay, so it's confusing because he says the mesoderm is filling up in the middle and his directionality is going from the head down. And that's interesting. I'm just wondering if that's important.
Because the red is in the front, you mean, from the head down?
Yeah,
Yeah, I don't think he's specific about where it starts.
okay, good.
Yeah,
So it's coming in between.
yeah, so there's that dip in the split, and then there's a split between the 2 halves in the top. There's she's talking about that.
Ectoderm, the underneath black, which is the endoderm, and then we've got the mesoderm now filling up in the middle, which you can see here as well. So it's coming in between from the epiblast in.
Okay, there's your answer. It's coming from the epiblast in, and that's that open space. It's, I assume, is what he's talking about. Is that okay?
Okay,
You know, I've
Okay.
never heard the word epiblast before, so I was going to go into Netter and try to find it in a different section. And honestly, I'm not. Because I have the same question you do. I've never seen a space created. I've only seen solids that say, hey, I'm a nervous system ectoderm. This is mesoderm, this is endoderm, endoderm being the gut side of things.
Mhm.
And because
Epiblast and the hypoblast, you remember Bonnie talking about that?
no, I don't. You wanna?
Here, can you see this? Can you see what I'm seeing? I put up.
Yeah.
epiblast
I'm
is-
reading the first sentence, but there is nothing more than that. It's embryonic development. The epiblast is one of the 2 distinct cells layers arising from the inner cell mass,
Yeah, from the blastocyst.
from the blastocyst. Oh, I see. So then can you move it further just to see the
Yeah.
photo?
It goes to, yeah, I will, just, here we go.
Okay.
In amniotic embryology, the
gotcha.
hypoblast is one of 2 distinct layers arising from the inner cell mass of the blastocyst.
So that's after the implantation. So the red thing is moved in already, right?
Well, no, this is right in the beginning. So this is before, this is as the egg is fertilized, it travels down the fallopian tube.
Gotcha.
There we're looking at the development of the embryoblast into the epiblast, the hypoblast, and the trophoblast. That's all happening in the first week. If I remember correctly, first 2 weeks. That's before we get to the 3 layers. In the beginning, the epiblast is one of the 2 distinct layers arising from the inner cell mass of the blastocyst. The blastocyst becomes the morula. So,
And the morula implants through the back.
through the back, the morula implants as it passes out of the fallopian tube and travels the ocean of the uterus and implants in the wall of the uterus. When you say the back, I'm not sure what you mean, which
Oh,
back?
I'm just saying that it's the back that will become the amnio. It's the Bonnie always says that we enter the mother through what will become our back body.
Yeah, she does. That we nestle and we do this. We nestle backwards
Yeah.
into
Through the back body, yeah.
Okay, let's see where we are now. All right, are we clear?
Yeah, so far.
Okay.
Clear enough.
Yeah.
Do you remember hearing the epiblast? Not Sylvia?
Not really, but I'm okay about keeping on going.
Okay, just trying to remember, if you look at the embryology in the first week. Let me look at this again.
Yeah, I'm just trying to,
During the second week of human
Okay.
development. The cells spread into a flattened tissue layer and differentiate into a two-layered tissue containing the epiblast, which is the columnar epithelial cells, and the hypoblast, which are the cuboid epithelial cells, which are together known as the bilaminar embryonic disc.
yeah, and I'm looking at another netter picture here.
Okay,
And it shows that that space of going up, in his picture, he's actually showing the transfer of cells, and it happens around the primitive node and the primitive streak. So if I look at another
what page
netter
are you on?
picture,
What page?
in some ways that little entrance that he drew there,
But what page are you on? Are you looking at the video now?
well, his picture in the video figures into something called gastrulation, which is page 36 of this version of Netters.
Mhm.
Um,
That's the forming of the stomach and the intestinal tract, etc.
well, it's more than that, but basically where my finger is here is equivalent to that. That little bit of red there that's shown there is the primitive streak. Yeah,
You're talking this? The mesoderm?
yeah,
He didn't even say primitive streak.
he doesn't, but that's, now I can track it because
Essentially the
Yeah, go ahead.
I have played with that, so that makes sense to me personally. And that transition from one layer to the other has to do with that point. And that's that little entrancy thing there is at the primitive node.
This entrance?
Yep.
Or
If
how
you
about
go
that
back to
little
page
circle?
36 of Netters,
Lydia, do you have Netter's book?
you have something similar. So this little place of the primitive node is similar to that bump up that then has the transition set of cells that are going through.
You're on plate or page 56?
36,
I see.
Three.
Yep,
36.
36, page 36. I'm on 36. And I see ectoderm, notochord, primitive knot. I see the primitive knot. You think that's where that little drawing is? Huh?
Yeah. I think we're at that stage of stuff, the beginning stuff of us.
Yeah, I think that he's drawing that notochord in red, unless we're just he called that the mesoderm.
Which it is. It's made out of mesoderm. It's the beginning of mesoderm.
Okay. We read out loud the gastrulation process out of Netters. It says it's the production of the intraembryonic mesoderm from thickening of the ectoderm, the primitive streak and primitive knot. The latter, the primitive knot, forms a midline cord of mesoderm. Okay. The notochord. Oh, did we lose?
Oh, we did.
She wasn't sure she could come in because of the storms and she had problems getting in. She sent me a message that she wasn't going to be able to come, but she got there.
But that
Back, yeah. So let's go on, see what happens here.
I was just wanting to be in the same type of language, and now I'm able to go, okay, I understand this.
Okay, that's good. I'm glad. It's always helpful to know what they're talking about. Very nice. So thank you for clarification. And we'll go on and we can review again when she gets back on.
Oh, there's Lydia.
Oh, she's back on?
Well, I see her name. She might not be able to have her visual. Yay, welcome back.
I'm not seeing her.
Well, I get to see her. She's holding up her little cart of
Yeah, sorry,
biscuits.
I'm here. I'm here. I just lost you. My line was not clear.
Oh, fabulous. I'm so glad you're back.
Thank you.
I am not seeing your face at all, so I need to back out a bit. To see if I can't get you. So I'm gonna stop the share and come back. There you are. Bravo, bravo. So with the help of Netter and Sylvia's insight, we got to discover that drawing, we'll go back to the drawing. Now, can you see the drawing?
The epiblast will make fibers that are going to come down. This is again a sad.
Do you see
Not
the,
shared. Oh, I see your drawing. Okay, gotcha.
you see his?
This same place is what he's talking about, and this is more the visual orientation I'm used to thinking about in terms of myself.
So this might be a little brighter to see,
Uh-huh.
Oh, that is.
right?
Okay.
And you can see, I can't hold the book and point to the structure, but we'll
time.
see the worm, the red worm. and-
Oh, I see. And it says primitive node.
Yeah.
Okay, so that little bump is a primitive node. And
Yeah.
at the bottom on his drawing, it's a connective stalk. Okay.
Yes, he did not label it. We're so there, yeah, we are a little ahead of the game there with the names.
Yes.
So you were very nice to pick up that opening in the center, which is what we discovered to be the primitive node, correct?
Yes. Not node leading to primitive streak leading to notochord,
Okay,
depending on which day.
okay. Here comes Andrew. So I'm gonna make a little note that we have. Andrew. I'll give him a second to tune in. Hey there, Andrew. This is like the four of us, so we're gonna let you in on what we've been going through. I found a nice video about the development of the first couple of weeks that would help clarify things for everybody.
Okay.
And you know Lydia, and I think Lydia, this is Andrew,
Hello, Lydia.
and you must know Sylvia.
Yes.
Okay.
Hello.
So we'll look back at this beginning of we're talking about the development, and this guy is there's a Dr. Matt and Dr. Mike. Can you see the page?
Not yet. You haven't screen shared yet.
Okay, let's do that. Now you can see it?
Yeah.
It's
Okay.
black. Yep, now it's there.
Okay. So Andrew, just to get you oriented, we're looking at the development of the nervous system. And he's just talked about the 3 layers, but we'll
Yes.
back up a little bit.
The mesoderm is now filled up the middle. So you've got your 3 of the neural tube. And then
Okay,
finally,
we'll start.
briefly, the peripheral nervous system. The rest
Okay,
of the
let's
lecture,
start
we
with
will
you.
look at the general development of the central and briefly the peripheral nervous system. There are kind of three main learning outcomes for this particular lecture, and the first being the formation of the brain vesicles and the folding of the brain itself. Number two is how the neural tube forms and causes the creation of all the cells in particularly the central nervous system. So that's almost more like histology of the neural tube. And then finally, we'll finish in number three, being the positional change of the spinal cord itself. So let's go across to this image. Now, these two images we used in the introduction of embryology or the embryogenesis. So if we look firstly here, now this is about 3 weeks. We've got the top blue, which is the ectoderm, the underneath black, which is the endoderm, and then we've got the mesoderm now filling up in the middle, which you can see here as well. So it's coming in from the epiblast in, essentially the
Okay, so that started our first questions, just so you know, which was what's the epiblast? And the epiblast is what helps form the 2-layer disc, the bilaminar disc, which precedes this 3 layers.
Mm.
Okay, so, and we discovered that because Lydia asked us what this little opening is here. We discovered it through Sylvia's exploration of netters that this is the primitive node here.
Mm.
Yeah, and this is actually going to be the primitive streak.
Oh, wow.
Yeah, so that's what that's about.
Sarah, where is the primitive streak?
4 layers,
Um,
but in this case, we've got the
primitive
3
streak
layers
will be
in
here.
place now. So the mesoderm is
Okay, so that is what it's confusing.
Uh-huh.
I hate to do this, but you see in the picture, I don't know if you can see it. The primitive
He says
streak,
the mesoderm here. Yeah.
yeah, the primitive streak arises on the end where there is a connective stalk. That's why I'm surprised, because it rises up from the bottom, which will be the pelvic floor perineal body. So it
Right.
should be
And you see
arising
the
from
ring. You
there.
see the ring. Yeah. So
3 germal layers intact.
yeah, so it may be confusing for us, but if we look at the netter's description, we do see the words. Let's see. Can you see this? Can you see this? No. See, let me come back to stopping the share. It says in netters, a migration of cells from the primitive streak to form the intraembryonic mesoderm. So
Yeah, and where is the connective stalk?
yeah, the connective stuff
On
would
the-
be somewhere in here, because here's,
Yeah,
yeah, so he doesn't go into that detail.
yeah. So it looks like the rising of the primitive streak came on the side where is connecting stalk. That's why I'm confused that this gentleman has the connective stalk on the other side. But that's okay.
That's caudal. He's got it on the caudal end, does he not? Did I miss that?
Yeah, well, one of the things I'm looking at it, when we start the primitive streak, it goes up, it gets to a certain point, and then the notochord kind of goes both directions.
Right.
The first sets of cells that create the primitive streak, which is sort of just a vertical line of force, they come from the bottom edge. But when I look in all the other netter information, what we think of is called cloacal, meaning the base of us here. It's just a tacking or an indentation of cells. And this part that he's talking about is above. And when I look to later images of what's going to happen in terms of connection, there is there's some other things that happen in terms of differentiating where is tail and where is belly button. So
Yeah, that's later. I'm just talking about the connective stalk. You see the
Yeah.
first picture? So the connective stalk is here, and this is where the rising of the red. Let's use the colors that Bonnie talks about. So the primitive streak rises here, and then there is a primitive node. And then, as you said, it's going in both directions. But on his picture, the connective stalk is here, and the red is here, and here is this space. So that's why I got confused, but that's okay. We can keep going. I'm just
That's good to,
confused with the
to know.
directionality.
Yeah. I think that he's just trying to streamline it, so not as detailed as what we're thinking of. Good for us, right? Good for us to be so detailed. Okay, let's get him back. So here is the caudal end. Here he's got a little loop around it, right? I don't know
Which would be a cloacal membrane, right? That would be the cloacal.
here would
Yeah.
be, yeah,
Yeah.
Yeah, cloacal there and oropharyngeal at the other
oral-
end, yeah.
oropharyngeal at this end.
Mhm.
Okay. All right, so are we on board? This becomes a primitive streak. I don't know if he goes into that. Let's see what he says.
Now, this is cutting through in a cross section, and if you were then to fold it back and look down, that's the view we've got here. So we've got the endoderm underneath, endoderma here, going down to the yolk sac, going down to the yolk sac, and then the ectoderm reflecting up like so into the amniotic cavity. So
Is that okay?
Mm-hmm.
towards the end of the third week, we've created the mesoderm, but what happens down in the midline, right down the middle of the embryo, is this big bar of tissue known as the notochord. The notochord gives the symmetry, should I say, of the embryo. So that will allow it to have a right and left side and go right down the middle of the embryo. Now, this notochord, in about the third week, will start to cause a change in the thickness of.
Okay, so he's clearly not saying where is the notochord coming from.
No, he's
No,
not.
he's not as detailed.
Yeah.
That's okay. We are.
Okay, yeah, he left out.
Very important point.
What? Okay, here we go.
The ectoderm right adjacent to it. So right behind it, it starts to cause this really undifferentiated growth on the ectoderm like so. And that's going to cause a neural plate to form. So that's a big thickening to form. Now that thickening, as we'll move up to this image, will cause the cells in that region to start to fold up like this. So this is the beginning of the neural, the cresting, or it's kind of cresting up or folding up. So this is the neural fold coming up into like a wave. So you can see it cresting up like so. The rest of it will continue across like that. That's the amniotic going up like that, and that's.
So while he's talking, if you want to go through the embodiment part of it, where you're folding it, can you see me or not?
I can't see you right now.
Cannot see me. Okay. While he's talking, you remember we did, in my class anyway, we did a whole folding and then embracing of the becoming the neural tube. So please go ahead and feel it in your body, even though it's intellectually, he's describing it, but let's take time to feel the folding if you want to. So do you want time to do that on your own? You want to do that while he's talking?
It's okay. I mean, we can just have him speak and pick up on what.
Yeah.
I don't know why you have him particularly, but
It was just by chance.
okay.
By chance. I like the way he was
making things clear. The other ones were, like an hour long, so we're not gonna go there.
This is the top layer, like so. So all you need to know at this point is around the 3rd week, the ectoderm, due to the influence of the notochord, will cause not only the plate to form, the thickening, but it will start to cause the plate to start to fold up like a wave, okay? Now, if we go across and look at this image, what we'll see, what you need to get your head around to see this image is here is the head end, here is the tail end. So this is again a sagittal cut. So it's like the embryo's head's here and the tail's at the back end. Now, if I was to look down on the embryo, this is the view you would get. So you're looking down on the embryo like so. So this is the amnion. This is all the amniotic cavity or sac folding to close it all in. So where it's starting to crest up and almost close like a circle is happening right there. And this is where the tube will finally close. And so these are cells around like that. So it's an enclosed tube, completely surrounded by these cuboidal shaped cells, which are the neuroepithelium. And where that first closes is in the cervical region of the embryo. So in this region of the embryo is where it first closes. And that's at about day 21, okay? Now, what will happen here is this closure will start to zip shut both cranially and zip shut caudally. So that will start to close, 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 and close, close, close on both directions. Now, as it starts to zip shut, there's 2 ends that are left open. This allows the communication between the neural tube in with the amniotic cavity. So it can kind of cycle through with the fluid or the amniotic fluid or the future CSF that's going to be made in that tube. Now, the top end, before it closes shut, is called the cranial neuropore, and the bottom end is called the caudal neuropore.
Okay, do we have any questions at this point?
Yes, I just have a question. If that zip, middle, is that where it first starts?
What he said.
Behind the primitive node is the neurotube is starting to close in the center around the same level with the a primitive node, and then going up and down, or what is the directionality of the closing of the neural tube?
My experience was I feel it is from the sides in, so it's pinching in, so to speak.
At the same time everywhere, or is it maybe a spot that starts first in the middle and then you don't, there is no, okay.
Starts first in the middle. That's how he implied that, the description.
Oh, okay, gotcha.
The question becomes here, he talks about, I thought he said cloacal when he was talking about this closure. His accent a little bit, I don't follow accents very well sometimes, so I'm not sure if he said cloacal. We can go back and look at that.
Here is this closure will start to zip shut both cranially and zip shut caudally.
Okay.
Folding to close it all in.
What? You have a
So
question?
where it's starting to crest up and almost close like a circle is happening right there. And this is where the tube will finally close. And so these are cells around like that. So it's an enclosed tube, completely surrounded by these cuboidal shaped cells, which are the neuroepithelium. And where that first closes is in the cervical region of the embryo. So
what word is that?
Cervical.
Cervical.
Cervical, because the early somites,
Yes.
okay, go back to Netter, and I'm on page 53. The occipital and the cervical somites are basically where the closure happens.
Mm.
First pictures where the somites, on page 53, it shows 3 occipital somites, and then the second to the bottom, day 21, it's showing more of cervical somites. So I know, yes, exactly.
Bring that closer up, Sylvia, so they can read it.
Yeah, so I was kind of looking at it. So in some ways, as we crest ourselves backwards, we're cresting ourselves backwards in this area of what will be our body. And I find that helpful.
Let's take a moment, guys, to feel the crest cells coming back and forming the somites here. The first, I
Which means somites mean mesenteric little middle red layer travels through and helps create this formation of closure.
Okay, where did you find that information?
It's because Bonnie, when she did her drawings, she did, I'm gonna modify Netter here. And give me a second here. But when she drew it, the somites were red.
yes.
Does that make more sense then, Lydia?
Mm-hmm. So the somites came from the mesoderm, and then the tongue and the first 3 vertebrae, as well as the muscles of the one that she was talking about, all came from those 3 somites that are up here.
Yeah.
Okay.
Actually, four.
4, okay.
Yeah, there are more somites, but asomite means I'm a mesenteric layer of cells migrating, in this case, coded by nerve cells.
Mhm. Yeah.
Yeah, we have a lot of.
Okay, so the first
And
folding is
what I just like about coloring something is that it makes it simplistic again, so then I can understand it.
And what was the
But that's
first one?
great. So our first folding was right behind our neck
Yes.
and kind of
Or at the very base of the very base of our occiput. The first folding is, it's like if if I do this, right? So I'm growing up the cells and then I'm closing and then I'm allowing the vertebrae bone structures to come through.
What if we look at the picture that that guy has about, he talks about the folding at one point, so.
Okay,
Mhm.
go back to your video. And it's very helpful, like, oh, if you just held someone's nervous system right at the back of their head and neck, that must be why it's so satisfying to have body work done here.
And I just want to make a point that it's not just the tongue, it's the muscles of the tongue. So the muscles of the tongue, and I haven't verified this yet, but they must include the infrahyoid and the subhyoid muscles, and probably the omohyoid. I don't know if it goes to the omohyoid.
Yes, good
But-
point. Thank you. And then trapezius and a sternocleidomastoid.
Yeah,
I don't know if they derive from that same area.
yeah, the sternocleido does. I
Yeah.
remember.
Bonnie said those group in trapezius and sternocleidomastoid are coming from the 3 somites in the same section.
Okay, that's good. That's good. I got to thinking about that because I spent a lot of time thinking about the tongue, working with the tongue, so that's very important to see that it's not just the tongue, it's the other muscles that are
Yeah.
around. too.
Right on.
I mean, okay, so here we go.
In this region of the embryo is where it first closes. And that's at about day 21, okay? Now, what will happen here is this closure will start to zip shut both cranially and zip shut caudally. So that will start to close, 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 and close, close, close on both directions. Now, as it starts to zip shut, there's two ends that are left open. This allows the communication between the neural tube in with the amniotic cavity. So it can kind of cycle through with the fluid or the amniotic fluid or the future CSF that's going to be made in that tube. Now, the top end, before it closes shut, is called the cranial neuropore, and the bottom end is called the caudal neuropore. Now, eventually it will close. It will close shut firstly up here. Now, at about day 25, it will close up at the cranial end, and about 2 to 3 days later, it will close down at the caudal end. Now, where it closes cranially, if we go across to this image, so this is the neural tube here, like so, where it will close is about there. And so that's in the actual adult now, it's a structure known as the lamina terminalis. So that's kind of getting close to the pituitary gland.
Isn't that interesting? 'Cause
Yeah.
Yes, but
the way that
cool.
this is described.
What I think it's cool also that he's not mentioning is that on the day 21, 22, it's also the beating of the heart. So basically, when this closes, we have the above the heart was the diaphragm, and then folding of the heart back in. The same day is the closure of those somites of the neural tube.
Yep. The first closure of the neural tube. Huh, echoes first beating of heart. Okay.
Yes, yes, that's well put. Wow, that's beautiful.
But I'm not sure if that's true. I'm just gonna look for it.
Guys, you have to realize this is just a recipe that not everybody follows. So your rhythms may be very different from the rhythms here. And this is a kind of a general picture. So if it's right for you, it's right for you, but it may not be, it's not the end all be all. It's not the, you may be a day before, you may be a day late. That's my understanding.
Yeah, but we're talking about only a day or so. So it's definitely that there is some relational coincidence there.
I don't know if that's possible because at 20 days, there are just blood islands. Which is page 93.
Then the 22 day, it's the starting of the beating of the heart. Some
Oh, it
say
happens
21.
very fast. Okay, that's wild. We go just from having a little bit of, primitive streak, notochord, having plates. Couple days later, we have blood inhabiting us. And then the blood coalesces and starts having an opinion about what to do very quickly. That's cool.
I love how you put
Okay.
the wording in. Sylvia, really like your wording.
Well,
Lovely
Mm,
wording.
thank
you have.
you're welcome. I'm
Yeah, let's take the folding.
yeah. Okay, so Sarah, later on, and for whoever who's looking at this is Netter page 95. And I'm going to just say that this is the neural tube from this perspective, this gray structure.
Okay,
Oh,
so
I see.
that's- I'm gonna make it a little clearer. I don't know why yours is not.
Yep,
For me, it's
One,
just
I don't know why mine isn't clear either.
The light, I'm facing the
what's-
light.
What's wonderful about that is that you can see that everything's tubular. We're plates, cell plates that have gone into tubes. And then these tubes, as they get more complicated, you're saying that at that point, we're gonna start to have this cell heart beating thing. And this is when the right and the left sets of blood flow come together, and they're coming together here. as the neural tube is growing this way, the front of the neural tube closes, and I love that palm there to be able to give me a lamina terminalis. And here I'm doing this.
Okay, so
Mhm.
But
let's
all
do
of
that-
me
let's
is
do
just
that as a
little
group,
tubey
Sylvia.
bits.
Let's do that as a
Yeah.
group. So I'm gonna feel, for me, I like to feel the rising of the heart
Okay.
coming up and leading that fold
Mhm.
over the developing brain. And that brings me into my hands and into prayer position. And then as my brain develops, I feel the reverse positioning of the brain cells coming back. That would be the cerebrum. So there's a fold and then an unfold for me. And then I'm at the lamina terminalis, feeling the cauliflower of the cerebrum, dividing into the sulcus, the longitudinal fissure rather, the left and right halves of the cauliflower. And I'm thinking lamina terminalis into the cauliflower. And then I start to think about the septum pellucidum in the center, dividing the cerebrum, which brings me into the space, the cave of the septum pellucidum, which we talked about last week and the week before. I think we talked about the septum pellucidum.
Please remind me, I wasn't there. Just a
That
space
is a
of it.
little triangle of tissue, the double membrane, which gives the midline. It's the basis of our midline, which we know is so important in development. That comes from this, the double membrane of the septum pellucidum.
And it's knitting right above that. So then we will have the hypothalamus and the pituitary right above that. Is that?
No, I think it's forward of that and a little lower than that.
Mhm.
The pituitary is lower and forward, so, and then if I pretend, because we have to go back into the brain, if I take a point to be a pituitary and this to be the front of the lamina terminalis, as I go back from there and I find the structure, that's where the septum pellucidum is. And it's an open, slightly fluid-filled structure that's different than the oceans of the interior of my brain. And the oceans are fronted, topped by the lamina terminalis.
Hmm.
I did go back and look more about the fluid, and it does originate apparently with the ocean.
Yeah.
So,
I'm just looking Bonnie's drawing right now, and I see the septum transversum that is above the heart and then going down and supporting the heart from below. But I don't see septum pellucidum. I'll take a look at it.
show you where the septum pellucidum. I'm going to show Bonnie's drawing because it's impossible to do without it.
Mhm. I
It's the
see.
orange on that.
Okay.
It's in orange.
Oh.
And the angled streak coming down from it, and then there's a knob at the bottom. That's where the pituitary is.
Yeah, pituitary is all the way down to the bottom in the front. But I
That's
thought this
why-
is when insula sits. I'm totally off then.
You told this was the insula?
No, the orange is the insula under the corpus callosum and the cingulate above.
This is medial to the, this is the deepest part of it. So you're medial to the insula.
Medial to the insula.
On the
Okay.
inside, the insula isn't, the insula would be out here. And
Uh-huh.
the rest of the brain would be out here. So then you go to the next layer, you'd have the insula. And then if you go to there, this structure almost disappears after, what do I say, 3 months?
Okay, so when you said it's medium to the insula, it might have been that the insula is sitting kind of on top of it then?
No, on the side,
Onto the right or the left?
both sides. We have an insula on the right and the left.
Mhm. And that would be in the center.
And the pellucidum would be in the center. That's my understanding.
Wow, thank you.
What she says, the septum pellucidum is a thin triangular vertical double membrane containing both white and gray matter. It is located in the midline of the brain between the anterior horns of the left and right lateral ventricles. It extends downward from the corpus callosum to the fornix, the hippocampus commissure. During fetal development, there is a space between the two layers called the cave of the septum pellucidum. It usually disappears in 90% of people during infancy. This space has also been referred to as the terminal enlargement of the spinal cord. It plays an important role in the development of midline, especially between the special sense organs of the face, mouth, nose, ears, and eyes. So we used that information to find our septum pellucidum and then find our midline when we did a practice with Bonnie, moving our hands towards the midline. And then you can use that to play with your senses. So you have this in your account, this drawing.
Yeah, I'm just looking at it. It's almost like a vertical, immediately inferior to it would be mammillary and pituitary and
Here's
hypothalamus.
the pituitary,
Mhm.
and your mammillary bodies would be somewhere here. And then your pineal would be up here.
No, down.
Oh, no, I think it's all much more down.
Yeah, they're all down from it.
They're all
Sarah.
down.
But you're using a pencil in front of a screen in such a way that
Mhm.
could be totally fussing with your ability to find these things.
Yeah,
But-
because what you're showing is the thalami, yeah.
Put your finger
Yeah.
on the line.
Okay, so here, pituitary. Here, septum pellucidum. That is the corpus callosum. This is the front of the,
Hypothalamus?
Lateral
No,
terminalis.
no, this is
Down.
lamina terminalis.
Mm-hmm.
It's incredibly difficult to do because I'm trying to turn around while holding it
Oh,
vertical for it not to, okay, so let's see if I can read it.
Yeah, we can see.
Pituitary,
here's the angle.
lamina terminalis,
Yeah, but let's go to.
corpus callosum.
Uh-huh.
This is the septum pellucidum. This is, what's that name of the green thing that was on Bonnie's picture?
Fornix. That anterior.
Fornix.
Sure.
Yeah.
It's the fornix.
This is the anterior commissure. That light bit there. One of these back here is the pineal. One of these two, and another one's the mammillary, and I'm not sure. So not looking-
Mammillary is on that line, so right above the pituitary, but a little bit angled and below the thalami.
Right,
It looks to me that mammillary is right on top of where the actual midline is, notochord is ending, where the midbrain is folded in the old brain, right there. Sorry, I can't share her photo, but I'm just
No,
looking.
you can't, we need your fingers on this.
Let's see what he says, because he does mention, I don't know if he talks about the pineal, but I know he talks,
Oh,
he talks about the infundibulum, which is the stalk.
okay, wait a second. I looked at the picture better and now I'm gonna turn around. That is where the mammillary body is.
Yeah,
Okay.
right above the pituitary and slightly on the angle. Yeah.
Yeah,
So
Okay.
can you go over?
pituitary, mammillary body, pineal, anterior commissure, posterior commissure. Fornix is this portion. Corpus callosum is the upper one. And then the triangular space between the two that I'm kind of pretending to color in is septum
Yeah.
pellucidum.
Whoa.
So let's see if we can finish this up for the video and make sense of it in our brains, in our hearts, and our bodies. Let's see. Okay, we're gonna go back.
Just
to-
a heads up, if I disappear, there is a client that come in,
Okay.
but I will get a recording, okay?
Okay.
That's going to close day 25, day 27 quarterly. Now, this is highly dependent on temperature. So if the mother's too hot, so if she has a fever, or there's been some research to show that mothers who were to have a sauna in about day 25, the cranial neuropore failed to close. And what that would result in is that wouldn't close shut, and therefore the embryo would develop anencephaly, which would be fatal and stillbirth.
Isn't that interesting? I never heard that before.
I never heard of that before.
Wow.
I know the spina bifida, but I didn't realize that if the cranial pore didn't close, or if you took a hot sauna, or you had a fever, that would cause encephaly. I mean, I know encephaly is not good, but I mean, the hot fever.
Unfortunately, I did know that one of the ways of causing a miscarriage is a very, very boiling hot bath. But it
Mm.
has to be done early. So we're
Mm.
just at 3 weeks.
Best at 3 weeks.
We're just at 3 weeks. You
Yeah.
don't get very many options. You have probably a 1 week period in which to have this problem happen.
Uh-huh.
Mm.
Okay. We lost Lydia.
Unfortunately, she has great questions.
She does. She's on top of things. Thank you, Lydia.
Caudal neuropore, that's more dependent, its closure is more dependent on folic acid. So if the mother was deficient in folic acid, it probably wouldn't close or would have issues with its closure, and that would give different degrees of what we call spina bifida. So that's essentially how it closes. Just before it closes, this is just an interesting point to be aware of. Before it closes, as it's starting to crest up, a group of cells, just as it's closing, will migrate on either side to sit here, and then they can move further, and they are called the neural crest cells. We'll talk about them a little bit later. So we've closed the neural tube now, and so what we can do is move across to these final two images for the first learning outcome. So what we can see is the neural tube has closed here, and this is now moving in a sagittal, so another sagittal section through like this, but we're just focusing on the neural tube now. So this is the neural tube coming up, and it continues to go into the brain or the future brain. Now, what happens is, because we now have shut it at least cranially, what will happen is the tube can't get any longer or bigger in it. And so you need to make all the cells in your central nervous system. And so the only way you can kind of make more cells is to dilate it outwards. And so at about week 3.5, we get 3 kind of dilations, 3 primary brain vesicles, okay?
So you remember Bonnie talked about how we grow laterally,
Mm.
but from in our cells there from the brain. Where's my notes? But yeah, we do.
So we can come across here and we can draw that in. So the neural tube will effectively develop the brain and develop the spinal cord. Okay. Now in the brain, where we've had the closure of the cranial neuropore, there's three main vesicles or dilations start to form. And these are what we call the primary vesicles. The first one, the big vesicle at the top, this is called the procephalon. Procephalon. The one in the middle about here is called the mesencephalon. And then the last one, which is kind of here down, that's called the rhombencephalon. Now, the reason why they're called that, pro is before, mes means middle, rhomb is rhombus, so it's its shape. So they're the three primary vesicles or the primary dilations. So you got dilation 1, dilation 2, dilation 3, and then we go down into the spinal cord. What you can else see here is you can see this notochord coming all the way up. The notochord, which is this structure here, so in this section it's coming all the way up and ends just at this point here. What you can also see here is you've got kind of a growth coming out of the neural tube at the proencephalon. And so what's that going to be? That's going to be the infundibulum, which is the outgrowth of the hypothalamus. And what you can see coming in here, this is a kind of pouch coming from the black, which is the endoderm. So this is the tube of the mouth coming to a pouch here like so. And this is going to be the mouth of the baby. So this is the start of the endoderm. The endoderm makes the entire gastrointestinal tract. So the mouth all the way through the anus is coming through here. But you can see the pouch going backwards to meet the dilation going out. And this is actually the formation of the pituitary gland. So you've got, this is going to be the anterior pituitary gland, and the one coming from the neurotube is the posterior pituitary gland. And the notochord is where it finally finishes cranially. That's going to be the Turkish saddle, which is where the pituitary gland sits.
Isn't that amazing?
Yes.
It's so incredible.
Turkish salad, the saddle. Turkish salad, that's funny. Turkish saddle. Yes, here's the saddle. So it's interesting.
Sarah, I must leave. I just want to say thank you, and please send this to me, and I'll talk with you soon.
Okay, I'm going to, yes, thank you for coming, and I'm going to stop the whole class for now and pick up next week, which will be our final class, where we left off. Okay. All right, so I'm going to end us here.
Okay.
Turkish saddle.
Okay.
And hop on your Turkish saddle and go for a ride.
Yes. Yes.
And it was fun to have you here. So thank you for coming.
Yes. Thank you. Thank you so much.
Great to
I'll
have
see you soon.